RNA purification from blood samples. During the next step, there is homogenization. Once we have got the palette of the white blood cells, then the next step is the homogenization. Homogenization is by adding the trizol solution to each of the tube and the cells they are resuspended in this solution. Homogenize and lyse the cells. Incubate for 10 minutes on the ice. This process is called as homogenization. So those white blood cells or the nuclei of the white blood cells that we got during the previous lesson, during this lesson, now we are going to homogenize those cells or those nuclei by adding the solution. The name of the solution is trizol. Once homogenization is completed, then the next step is phase separation. So add chloroform according to the amount of the blood and vigorously shake or vortex each tube for 15 seconds and lets the sample stand for 10 minutes. As a result, there will be two separate phases. One would be aqueous phase and one would be organic phase, because we have used trizol or we have used the phenol chloroform. As a result of these solvents, there should be two separate phases. So that is the reason this step is called as phase separation. So we have separated the phases. Centrifuge the sample at approximately 14,000 rpm at 4 degrees centigrade. So during this step, we have separated both the phases, the lower phase and the upper phase. Upper phase is aqueous phase and the lower phase is organic phase. During the third step, there is RNA precipitation. So once both the phases they are separate from each other, remove the upper phase and transfer to a clean microcentrifuge tube, because RNA is present in the upper phase. So upper phase, all the solution which is shown as upper phase, that is removed and transferred to another tube. And once that is transferred to another tube, equal volume of the cold isopropanol is added. So when we add isopropanol to that solution, there is precipitation of the RNA. While doing this process or performing this process, be careful that do not take any white interface which was present between the two phases, the lower phase and the white phase, because in the white interface there can be the DNA. So just try to take the upper solution, that is a white aqueous solution. RNA precipitation: immediately vortex and place on the ice for 10 minutes. Samples can be centrifuged at 14,000 to 17,000 for 10 minutes. So in case of RNA precipitation, these are those steps which are performed once we collect the nuclei of the white blood cells. Once we have nuclei of the white blood cells, then the next step is homogenization. Once homogenization is completed, then separation of the phases, both aqueous phase and the lower organic phase. And once have separated, samples should be transferred to the next tube, where by adding the isopropanol or ethanol, RNA can be precipitated.